All this complex biochemistry of nutrient molecules and cells is just the endpoint of a longer sequence of events relating to processing of food, energy, and nutrients. Those involve complex systems as well, with complex patterns of control and optimization. Let's first lay out a little vocabulary. We start with food, which we're going to define as energy-rich materials taken from other creatures. This is what heterotrophs do, and animals are, without exception, heterotrophs. Food includes the various classes of chemicals we discussed earlier, but now on the hoof, so to speak: carbohydrates, proteins, and fats wrapped up in the package of a functioning organism that presumably doesn't want to give it up to nourish another creature. Once taken, however, food must be converted into nutrients, the constituent compounds that make up the various classes of foods. This includes conversion of storage and structural polysaccharides into monosaccharides and disaccharides, conversion of proteins into amino acids, and conversion of fats into triglycerides and fatty acids. All of this takes place outside the body of the animal proper. Before it can do any good, nutrients have to be taken up across the boundary that separates the inside and the outside, which, in the case of animals, is the intestinal wall. In other words, all the nutrients have to be absorbed across the intestinal wall to inside the body. The process of digestion refers properly to the conversion of food into nutrients, that is, the breakdown of the complex molecules that make up the body of the creature being eaten into nutrients that can be absorbed across the intestinal wall. We're going to take a broader view of digestion and include in it not just the chemical breakdown of carbohydrates, proteins, and fats, but also the finding, capturing, and rendering of the packages, so to speak, the organisms, in other words, that are giving up their substance to the animal that's eating them. Digestion takes place within a digestive tract, which is topologically outside the animal's body. There are two types of digestive tracts to be found among the animals. The first and relatively rare type is the incomplete digestive tract. Digestion in an incomplete digestive tract takes place within a blind-ended compartment known as a gastrocele, which opens to the outside through a single opening which serves as both the mouth and anus. This type of digestive tract is found only amongst a couple of primitive animal phyla, including the Cnidaria, which includes the hydras, corals, sea anemones, and so forth, and the platyhelminthine worms, or the flatworms. The most common digestive tract among the animals is the complete digestive tract. Here, there's a distinct mouth and anus with a long digestive tube that connects the two openings. Notice here that the digestive tube is still topologically outside the body. Usually, just inside the mouth, there's a widened area known as the pharynx. We've already encountered the pharynx in talking about the embryological origin of gills and lungs. Complete digestive tracts are found among nearly all the animals, but these arise embryologically in two distinct ways. In animals, gastrulation produces a gastrocele, which has only a single opening, the blastopore, to the outside. Thus, the embryologically primitive gut of all animals is incomplete. In nearly all animals, however, a second opening to the gastrocele opens up, essentially converting the sac-like gastrocele into a gastric tube with a distinct mouth and anus. Among animals, there are two origins of these openings. On the one hand are the deuterostomes, which literally means second mouth, in which the anus is the primordial blastopore and the mouth is the secondary opening. These include the echinoderms, the chordates, and the vertebrates. On the other hand are the protostomes, which literally means first mouth. In these creatures, the primitive blastopore becomes the mouth and the secondary opening becomes the anus. The protostomes include the arthropods, mollusks, annelids, and a host of minor invertebrate phyla. We're going to now reconstruct the typical process of digestion using the vertebrate digestive tract as our model. Many of the things that go on there also go on in other digestive tracts, such as those of insects, mollusks, annelids, and so forth, so we might as well use the vertebrate tract as our model. We're going to come back to the incomplete digestive tract a little bit later when we talk about digestive tract design principles. Let's start at the beginning with the business end, so to speak. What goes on there is pretty straightforward. Food is taken into the mouth where it's physically rendered from the creature that was eaten into a paste-like slurry. There's a fancy name for this, trituration. This is pretty much a physical process aided by rendering tools projecting from the interior of the mouth. Among vertebrates, of course, these tools are the teeth, but they can come in other forms in other animals. In the case of insects, these tools are made from hardened chitin, and in the case of vertebrates with beaks, these tools are made from hardened keratin. Physically, the tools come in three broad functional forms. You have pointy things that impale and pierce, knife-like things that slice things apart, and flattened milling surfaces that can be used to crush things. As this rendered food passes through the mouth, glandular structures around the pharynx, known as salivary glands, secrete water, along with lubricants and enzymes that start the process of digestion. Among vertebrates, one of the principal enzymes of the salivary glands are amylases, which begin to digest any starches that might be present. There are some vertebrates that do not have teeth or that only incompletely render the food taken in at the mouth, and this is true for other animals as well. And in this instance, further rendering is sometimes provided in another expansion of the pharynx, a muscular region known as the crop. The crop, if present, has quite thick muscles, and in some animals, the interior surface of the crop is molded and shaped into hard ridges. There are many animals that also include within their crops small stones that they have swallowed, known as gastroliths. When the food passes through the crop, its muscular action and the rendering surfaces of the crop, or the gastroliths, can render the food further. The rendered food then passes along to a tubular esophagus into an expanded chamber, the stomach. Here's where digestion proper really begins. There are several things that happen in the stomach, but mostly it produces a chemical brew that starts breaking down into smaller constituents, the large molecules in the bolus of rendered food. The major thing that happens is the secretion of a strong acid, hydrochloric acid, or HCl, into the mix, acidifying it down to a pH typically around 1 or 2. This has a number of important effects. In acid conditions, for example, proteins begin to denature. The quaternary, tertiary, and secondary structures of the proteins begins to degrade. Covalent linkages, such as disulfide bridges, are also broken down. This has the effect of causing the protein, which might have been tightly folded up, to expand and open up, which exposes more of the protein's peptide linkages for hydrolysis and digestion. Acid conditions also tend to promote hydrolysis on their own, so that peptide linkages and proteins either begin to break down spontaneously or they are rendered more susceptible to enzymatic breakdown. The stomach lining secretes a mix of peptidases, the most prominent being pepsin, and in animals that can digest milk, renin, which breaks down certain milk proteins. This continues the process of protein degradation, converting proteins into smaller peptides and amino acids. The stomach lining also secretes a small number of lipases, which are enzymes that begin to de-esterify fats into fatty acids and glycerol. This action is fairly limited compared to the protein digestion that takes place in the stomach, largely because the ingested fats are mostly in the form of large fat globules, which the waterborne lipases have a difficult time getting to. Polysaccharide digestion into sugars that began in the pharynx also continues in the stomach, aided mostly by the acid conditions of the stomach. And finally, there's a great deal of water that's added to this mix, which helps liquefy the bolus. All of this biochemical action is aided by the mechanical stirring of this liquefied mass of digesting food by the muscles of the stomach wall. And at the end of all this, what passes down the stomach is a liquefied digesting chemical stew known as chyme. Once the stomach is done with the chyme, it flows into the next segment of the digestive tract, the small intestine. There's quite a lot that goes on here as well, and we'll take it piece by piece. The big problem is that the chyme is very acidic, and it's filled with digestive enzymes that can eat everything up, including potentially the wall of the small intestine. The major thing that goes on in the small intestine is to change that acidic, corrosive slurry into something that not only completes digestion in the chyme, but readies the nutrients for absorption into the body. The small intestine secretes considerable water into the chyme to help dilute it, but more must be done, and there are accessory structures that help in this. One is the exocrine pancreas, which feeds into the small intestine via a duct, the pancreatic duct. The second is the largest gland of the body, the liver, which connects into the small intestine via the hepatic duct. In many vertebrates, the pancreatic and hepatic ducts merge before emptying into the intestine, but we're going to draw them separately here. The exocrine pancreas secretes several substances into the acidic chyme. Among the most important is sodium bicarbonate, which neutralizes the chyme's acidity and brings its pH close to something approaching neutral. This change of pH also tends to inactivate the pepsin and other peptidases that had been secreted into the chyme by the stomach, because these are structured to function well in acidic environments, and neutralizing that acidic environment also neutralizes their activity. Therefore, the exocrine pancreas secretes a new suite of digestive enzymes that function better in the neutral environment of the small intestine. These include a number of peptidases, of which trypsin is the most important. This completes the digestion of the originally ingested proteins into their constituent amino acids. The exocrine pancreas also secretes a third set of enzymes into the small intestine, lipases, which begin the digestion of fats into fatty acids. For these lipases to work, however, something has to be done to those large globules of fat coming into the small intestine from the stomach, and here's where the liver comes in. The liver secretes, among other things, a cholesterol-derived substance known as bile salts. These are emulsifiers, which means they act as soaps. Like your dish soap does with greasy dishes, bile salts convert the large globules of dietary fats into smaller and smaller globules until they're distributed among many small droplets of fat called micelles. These micelles are on the order of a few tens of micrometers in diameter, and here's where those pancreatic lipases can really get to work, because breaking up the fat globules into micelles gives these lipases a lot of surface area to work with. These lipases can also be activated by the presence of the bile salts in the micelle boundaries. What comes about in the small intestine then is the near completion of the conversion of dietary carbohydrates, proteins, and fats into amino acids, sugars, and lipid micelles that can be absorbed across the intestinal wall. We'll come back to the details of how these chemicals are absorbed a little bit later, but for now we need to carry on considering our process of digestion. The next segment of the digestive tract is the large intestine, or colon. What comes in here is a combination of the residual nutrients that have not yet been absorbed in the small intestine and a mass of low digestibility residue, which can include things like dietary fiber, roughage, and other things that digestion in the stomach and intestine have failed to break down. Within the large intestine, these residual nutrients are absorbed across the intestinal wall. However, the large intestine has a very rich microbial flora living within it, And these get to work on the low digestibility residue that the stomach and small intestine have failed to digest. This is useful because the microbial flora usually has a much more diverse metabolic toolkit to work with than does the animal itself. These break down the low digestibility residues to produce the conventional nutrients, such as sugars and amino acids, along with various volatile organic acids, such as acetate, propionic acid, butyric acid, and so forth, which are then absorbed across the intestinal wall. The microbial flora also produce a number of essential amino acids, various vitamins like vitamin K, other small complex organic molecules, and various cofactors like the B vitamins, which are also absorbed across the intestinal wall. Finally, there's quite a bit of reabsorption of salts and water across the intestinal wall, and this helps draw water from the low digestibility residue that's left to produce the relatively dry feces, which are then expelled through the anus. Well, that was an awful lot of material, but there is a lot of complex stuff going on in the digestive tract, and that was just an overview. There are several things we need to delve into more deeply, such as the function and anatomy of the intestinal wall, how nutrients are absorbed, how water and salts are absorbed, and how this whole thing is actually controlled in a coordinated way to get the best function out of this very complicated system.